Hello all. Today I'm going to show how to construct phylogenetic tree. Um, so let's start. So basically phylogenetic tree um is it's called evolutionary tree or tree of life. And the term phylogeny, it has Greek origin uh, and it is made up of two separate Greek words, phylon, which means tribe or species, and genesis is origin. So basically, phylogenetic tree depicts the evolutionary relationship among a species. So when we consider gene genetic sequences and protein sequences to derive genetic relationship among the species, then we generally use the term molecular phylogeny. We need to know important terms before we uh, learn how to construct phylogenetic tree. So this is a typical phylogenetic tree, and you see that it has nodes. So these are called nodes, and these are also external nodes. So external nodes represent actual organisms, and they are also called operational taxonomic units, O2U. Um, and these are internal nodes, and they are hypothetical taxonomic units. So these are hypothetical organisms. So this one is the root. So this is called the ancestor of all hypothetical taxonomic units as well as operational taxonomic units. And this is the branch or edge which connects this hypothetical taxonomic unit to the operational taxonomic unit, or internal node to external node. So rooted tree is basically a phylogenetic tree having a root, and root serves as an ancestor of all descendants. So these are all descendants, and this one is the ancestor of all descendants. Unrooted tree is a phylogenetic tree without any root, without any common ancestor. So these are also called, so external nodes, these are also called leaves. So in order to construct a phylogenetic tree, we need sequences. Either DNA sequences, we can consider RNA sequences, we can also consider protein sequences. So at least three sequences we need uh, to construct a phylogenetic tree. And those sequences must be aligned. So a sequence alignment and phylogenetic tree construction program is needed. So basically the step is you need sequences, then you align all sequences, you perform multiple sequence alignment, and then on the basis of multiple sequence alignment, the program will construct the phylogenetic tree. So here I'm going to describe a simple method to construct a phylogenetic tree, and that method is unweighted pair group method with arithmetic mean, that is UPGMA. So I'm going to use here MEGA. So MEGA is a software, molecular evolution and genetic analysis. It's a freely available software, and one can use this software to perform multiple sequence alignment, and using the molecule after multiple sequence alignment, uh, one can construct the phylogenetic tree. So let me first align the sequences. So for this, we have to go to File, Edit, Build, Alignment. So here I'm going to retrieve a sequence from file. So basically, you can create a new alignment. You can open a saved alignment session, and either you can retrieve a sequence from a file. So here I'm going to retrieve sequences from a file. So let me go to this phylogenetic tree file, and here we have I have 4 sequences, and these are actually amino acids. So this is short peptide MST RDF, sec2 is MTT RDF, sec3 is MTP KDS, sec4 is MST KEF. So I have here total 4 sequences. So basically UPGMA method is unweighted pair group method with arithmetic mean. So I will show you how this method works. So those 4 sequences are here also. So sec1 is MST RDF, sec2 is MTT RDF, sec3 is MTP KDS, and sec4 is MST KEF. And as we know that these changes, you see that sequence 1 is different from sec2, sec3, sec4. So all these sequences are not identical. So they can be similar, but they are not identical. Okay. So they are not completely identical. So basically these differences arise due to transition and transversion. So transition is actually mutation when purine is replaced by another purine. So basically when adenine will get mutated uh, into guanine, then it will it it will be categorized under transition. But when a purine will convert into a pyrimidine like adenine to cytosine, then it will come under transversion. So basically when these changes happen, then the codon will change, and you know that codon has 3 letters, and when the codon will change, then each codon represents an amino acid, so the amino acid will also change. So basically uh, differences in all these so these all differences basically denotes the genetic differences. So basically unweighted pair group method with arithmetic mean UPGMA. So here I will show you how to use how to calculate distances distance matrix, and how to use that distance matrix to construct the phylogenetic tree. So basically, in MEGA11, the program first calculates the distances, create a distance matrix, and then the program uses that distance matrix to construct the phylogenetic tree. So here, this p distance is actually it's a number of difference divided by total number of amino acids. So here basically it's when we consider a pair of a sequence, then we calculate the number of differences, means how many amino acids or how many residues they are different. So all sequences are 6 amino acids or 6 residues. They have 6 residues. So for example, if we calculate distance between sec1 and sec2, then number of differences are 1. So here you see that MSTRDF and sec2 is MTTRDF. So basically at second position, serine has been substituted by by the threonine. So there is only one difference out of 6. So the distance is 0.16. Uh, when we calculate distance between sec1 and sec3, then it's a total difference is 4. So the first difference is at second position, second difference is at third position, third difference is at fourth position, and the fourth difference is at sixth position. So you see it's 4 by 6, so that is equivalent to 0.66. So basically so the same way we calculate distance between all pairs. So all possible pairs are sec1, sec2, sec1, sec3, sec1, sec4, sec2, sec3, sec2, and sec4, sec3, and sec4. So total number of possible pairs 1, 2, 3, 4, 5, 6. So in UPGMA method, once we have these all distances between pairs, then we create a matrix. So it's a, you see, it's a matrix it's a where I have label I have sequence 1, 2, 3, 4 here in the column, and here in the rows, and here in the columns we have sec1, sec2, sec3, and sec4. So when we compare sec1 by sec1, then we will get the same sequence. So we have 0 p distance, and the p distance for sec1 and sec2 is 0.16. So in this way we fill this table, and we see that the diagonal is actually the comparison with itself. So only the upper part and lower part of the matrix. So the, both we can use either upper part or lower part. So what we do in UPGMA method, we first select a lowest p distance. So we see that the sec1 and sec2, it is showing 0.16. So the p distance is very low. It's a lowest p distance. So we group together sec1 and sec2, and then we create a new table where the sec1 and sec2 will be compared with sec3 and sec4. And in this way, now what will happen, how we will calculate the distance between sec1, sec2 and sec3. So sec1, sec2 is now a new group, and it will be considered as a single group combined. And now we have to calculate the p distance of this combined group with the sec3 and sec4 respectively. So how we will calculate that? So first we have to calculate the distance between sec1 and sec3, and then the distance between sec2 and sec3. And when we will have those distances, then we will have to take average of those distances. And this is the average of the distance between this combined group and sec3. So for sec4, we have to repeat the same. So in this way, we see that the sec1, sec2 as a combined group is showing p distance 0.41 with sec4 and p distance 0.58 uh, with sec3. It means like it is related, it is more related to sec4 than sec3. So we can group together sec4. So now we have new group where all these three sequences have been combined, and now we are going to compare with sec4. So we have this sorry. So here it should be sec4, and it should be sec3. Yeah. So basically, on the basis of p distance, we group here we have grouped sec1, sec2, sec4 in a single group, and now we are going to compare with the sec3. So it has 0.91. So sec3 is distantly related. So the sec3, it will be not combined. So basically here only two groups they have been left. So when you will have two groups left, it means like your calculation is now complete. So here what we got is it's a Newick format where you can see that sec1, sec2, they are they are in first group, and then the second one is sec4, which is close to sec1 and sec2, and then the sec3 is distantly related. So this is also a for this is also a representation of phylogenetic tree, because here based on the p distance we see that uh, sec1 and sec2 both are very close, they are closely related, and sec4 is also closely related to sec1, sec2, and the sec3 is distantly related to all this sec1, sec2, and sec4 sequences. So in MEGA11, let's see how to represent this. So first you have to select all and then perform alignment by either muscle or cluster W. Here I'm going to align by muscle. So you have to select default options. So this sequence has been aligned. This sequences have been aligned, and now we have to export alignment as MEGA format. So we have test.mega. So I'm going to override this. So just input title of the data is test. And now when we have this tst dot meg file saved, and then we can we can construct the tree. So in order to construct a tree, you have to click on Phylogenetic, and here construct and test UPGeometry. So UPG for this you have to open the MEG file. So this is the test.meg file, and here I'm not going to test the Phylogenetic, and basically the model is p distance. You can select other options also, but here I have shown how to use p distance table to construct the phylogenetic tree. That's why you have to select here p distance, and the rest of options you don't have to change. And then press OK. So this is the tree. So you see that sec1, sec2, they are very closely related, and sec4 is closely related to sec1, sec2, and sec3 is distantly related to all these three sequences. So basically this is here it is showing that it has root, so this is a rooted tree, but we don't know here this is hypothetical, and these nodes, these nodes are hypothetical nodes, but these nodes, external nodes, they are true nodes, and they are the actual sequences, and these are the branch. So basically the branch length represents the evolutionary distance, and also like it represents the differences. And here you see that basically this sec3, it has 0.40. It means like it is distantly related, but sec1 and sec2, they are very closely related. Here we can also show the distance scale here. And in this way, you can construct the phylogenetic tree, and you can also change the style of the branch. So you can also change so it in straight, or you can change it to curved, or you can show it as radiation. So this is also informative. Again, this branch length, it is showing, you can see this is like the branch length is very large here. So it means the sec3 is distantly related. So in this way, I like this rectangular style, traditional rectangular. And rectangular. And you can also save the session, and you can just you can save the image as TIFF or PNG file. So let me save it as PNG. So here tree test.png. So in this way, you can construct a tree. And let me construct another tree. And this time I'm going to utilize coronavirus, uh, spike protein of coronaviruses. So align, edit, build alignment, retrieve sequences. So I have collected 4 sequences of spike protein of coronaviruses, different coronaviruses. So let me open this file. So you see, I have edited the name, and basically these are 5 sequences, not 4, 5 sequences. So the top sequence is of spike protein from SARS-CoV-2, and we all know that the ongoing pandemic is actually for ongoing pandemic, this SARS-CoV-2 is responsible. Second sequence is spike protein from SARS, and this is the first version. Third sequence is spike bat rat G13. So actually, this is a spike protein from a coronavirus, which actually which is found uh, in bat, and uh, this coronavirus has a spike protein which is very closely related to the SARS-CoV-2. We have also spike protein from pangolin coronavirus, and we have the fifth one is spike protein from Middle East Respiratory Syndrome virus. So we are going to construct a phylogenetic tree from these sequences. So let's align. So we have to select all these sequences and then click on align by measure. Okay. So now all these sequences have been aligned. And then we export the alignment in MEGA format. So I have already this corona spike mega dot MEG. So here corona your spike, and then we go to phylogeny and construct and test UPGMA. So again we are going to use this p distance. So you see here that spike SARS-2. So spike protein from SARS-2, like spike protein from SARS-CoV-2 is is closely related to spike protein from RADG13. Okay, so bat coronavirus RADG13. So these two proteins they are very similar. Okay, like they are almost identical. Uh, and the pangolin is also closely related. Spike SARS is also closely related. But the MERS spike of MERS is is distantly related to this all uh, spike of coronaviruses and spike protein of coronaviruses. So particularly the distance between distance between spike of MERS and spike of SARS-CoV-2 is largest. So you can see, like, if we see the branch length here, if we go through all these branches, then it's distantly related. So in this way, if we try to validate it, like if we search literature, then we will see that the spike protein of SARS-CoV-2 is around 98% identical to the spike protein from bat rat G13. So let's search this. So spike. See, it's bearing 96% genome sequences. So it's yeah. So you see, this is despite SARS-CoV-2 and that G13 spike being 98% identical. Um, yeah, at amino acid level. So it means like this tree is correct because we see that the SARS-CoV-2 spike and bat rat G13 spike they are they are almost identical. Okay. So in this way, we can derive relationship between different organisms, and we can just guess that how the organism has evolved, um, and we can have like we can also predict that what can be the uh, future sequences like after after evolution. So uh, that I'm not going to cover, but like many evolutionary biologists, they use evolutionary tree a phylogenetic tree to predict the new species, and you can search it. Yeah. Well, the prediction is not reliable, but we can understand how this how a particular species is evolving. See this one, phylogenetic trees and the future of mammalian biodiversity. So basically, the phylogenetic tree is used to characterize also a new species. So whenever like those um, microbiologists, they when they have 16S ribosomal RNA sequence from a novel microorganism, bacteria, then they used to uh, generate. They used to compare they used to compare the 16S ribosomal RNA from the novel bacteria, and or then they used to compare it with other known organisms, other bacteria, and they actually they they generally establish the relationship, and they can find that where the novel bacteria fit. Okay, so you can see this. So basically, we can identify a new strain. So you can read such type of articles to understand the importance of phylogenetic tree. Okay, so you see this novel bacterial lineage and enzymatic potential of the alkaliphilic bacteria from the saline coastal desert of Little Run of Kutch, Gujarat. So in this way, basically, researchers they use phylogenetic tree to um, to establish the relationship between relationship of novel bacterial strain with the known bacterial strains. And also, yeah, so here I'm going to show third example where I will use myoglobin sequences to construct phylogenetic tree. So for that, file, not a session, rather, okay, so align, and it will build alignment, retrieve a sequence from file, and we are going to use myoglobin faster. So all 10 sequences, we need to align all 10 sequences, alignment by measure, okay. And then after alignment, we have to export the alignment in mega format. So myo, okay. Then click on phylogeny and construct and test UPGMA3. So here, yeah. So you see that now we have this phylogenetic tree, and we have 10 myoglobin sequences from 10 different mammals, 10 different animals, and or 10 different organisms. And you see that in this phylogenetic tree, Homo sapiens and that is human, and here chimpanzee, they have been placed together, and because the sequences of uh, myoglobin sequences, they are very almost identical, so, or very similar. So, and next is scrofa, and the myoglobin sequence of Ascaris, that is a worm. It's distantly related to human as well as plant troglodytes, and this can be considered as outgroup, and this is actually rooted tree, but here it is meaningless because you don't know which one is the root. But basically, in this way, we can see that which organism is closely related to which organism. Okay, so basically these protein sequences, they are showing the evolutionary relationship. Okay, so on evolutionary time scale, we see that the rat is this worm is very far from Homo sapiens. So it is like we can say, like, it can serve as ancestor of all organisms. So we can make it root. So generally, when you don't have root, then we select one sequence which is distantly related, and then we can consider it as outgroup, and then we can make that as root. Another is we can also test the tree. So let me show you how to test the tree. So in this, we have to, I'm going to use the bootstrap method. So here the number of bootstrap replication is 100. And you see that these are the digits. So basically it's like confidence level. And if it is 100, it means like it's most probable that the relationship is true. And if it is less than 50%, then there is no, like the confidence level is not very high. So in this way. And another, actually, the so bootstrapping actually construct the trees in various ways. So you will also get a consensus tree. Yeah. So save it as image, save as PNG. Okay. So in this way, we can construct phylogenetic tree from uh, using protein or protein, DNA, or RNA sequences, and we can derive the evolutionary relationship among the organisms. So thank you for watching this video. Don't forget to subscribe my YouTube channel